Greetings of the day. Myself, Annapoorni ammal, Assistant Professor, Department of Forensic Science, Kristu Jayanti College. Today, we'll be learning about the topic RH blood grouping system. Starting from the history of RH blood grouping system, in 1939, Philip Levin and Rufus Stetson had a case of a highly immunized pregnant woman. So a pregnant woman came to them, but the thing was, the child that was delivered was a stillborn. So due to this stillborn delivery, she lost a lot of blood, which was retained by giving her a transfusion from her husband. But later, it was seen that the serum of the pregnant woman agglutinated almost 80% of all the RBCs present in the blood group system. So they came to know that there is something called as ABO compatibility while transfusing the blood. They gave this topic to Karl Landsteiner and Alexander Wiener, who were already researching on the same topic of ABO blood grouping in 1940. So they did a research by taking some blood from the rhesus monkey. Rhesus monkey, which has blood, will be having some kind of antigen which will be different from humans and all other animals. They took this rhesus monkey's blood, injected it into the rabbit, or they took some small animals and identified that anti-RH antiserums was prepared in the body of the rabbit or the small animal. This antiserum was isolated from the animal and was then given back to the rhesus monkeys, which had some kind of coagulation in the blood, as rhesus monkeys had the same antigen to which the antibodies was produced from the rabbit. As part of the study, they took the same antisera from the rabbit and injected them into the human being. So they found out two different conditions, of which one, the antibody reacted with the RBC, and in few other samples it doesn't. So they found out and named this factor as RH factor, because this factor is mainly found in rhesus monkeys. Later they came to know that those human with RH factor can be called as RH positive, and out of the total population, 85% of the people are RH positive. And some of the human lack this RH antigen and they were called as RH negative, and 15% of the total population is RH negative. Coming on to the biochemistry of RH blood grouping system, the structure of RH is as follows. RH antigen is present embedded in the RBC membrane. It's a 12-pass membrane protein, which means that 12 times the membrane will pass through the structure will pass through the membrane. It is made up of proteins, hence it is having components of amino acids. So there will be each amino acid, and some of them will be embedded inside the membrane. The 12-pass membrane proteins is simply the basic structure of RH antigen. The antigen determinants are RH, D, C, E, and T will be attached onto the surface of this 12-pass membrane filter. Coming on to the detailed structure of this one, this is how it looks like when you go deep down into the molecular structure. So these round structures are called as the amino acids, and we can see that 12 times this passes through the membrane. And on the outside surface, we will be having antigens, that is mainly determinant of the RH blood group. Antigens can be either D, capital C, small c, capital E, or small e. Moving on to the types of RH phenotypes. If you have one RH antigen, there will be different types of antigens like D, C, and E. Coming on to the one single structure of RHD, it's as a representative structure. If you have D antigen, there will be 4 antigenic determinants. The 4 antigenic determinants make the D a complete good antigen. The first type of RH antigen or RH phenotype is when you have the proper D antigens ample amount on the surface of the RBC or embedded in the membrane. Such blood groups or phenotypes are called as D positive phenotypes. At the same time, if you don't have any D on your RBC structure, you are called as a D negative. If you have D antigen, but the number of D antigen is very less compared to the D positive, you will be called as a weak D. If you have D antigens, but if you see the number of antigenic determinants is very less, some will have 2, some will have 3, some will have none, then they are called as partial D. Along with that, one more phenotype is there, which is called as RH null, where no RH will be present in the RBC surface, neither D, C, or E. Such condition is called as RH null. Coming on to the genetics of RH blood grouping system, The first chromosome has a gene called as RHD gene or RHCE gene. These genes are the determinants which determines whether a person will have D antigen, C antigen or E antigen in their Rh blood group. RHD produces D antigen and RHCE codes for the production of C or E antigens respectively, and they are present in the position 1p36 to p34 in the chromosome number 1. Inheritance of Rh blood grouping. If you are having a particular Rh blood group, that is whether you are Rh positive or negative, is determined by based on what Rh your father and mother was having. Consider a scenario where the genotype of the father is D, which is homozygous dominant condition for D, and they are Rh positive blood group, and the mother is also Rh positive but she is having heterozygous D condition. The gametes that will be formed will have D, D, and small d. So, the first filial generation will have all positive blood groups, either homozygous D or heterozygous D. Consider another scenario where the genotype of the father is D small d and the mother is small d. So, the phenotype of the father will be Rh positive and mother Rh negative. With the gametes D, small d, and small d, the first filial generation will have equal number of positives as well as negatives. Blood transfusion and translation reactions. Whenever we give blood to someone, they will be taking the blood intravenously. So the process of transferring the blood or any blood products intravenously is called as blood transfusion. This is generally done to replace lost blood or blood components from a person's body. This is a blood bag in which we will be storing the blood while we give or while we donate for blood. So there are different parts in the blood bags. The main part is the blood bag itself where the blood will be stored, and on the blood bag there will be some blood bag labels which will have the details about the donor, the age of the donor, the blood group, the collection date, everything will be written there. Then there will be a set of tubings along with few clamps and needles and shields. There will be a sampling bag near the blood bag. So the blood bag is the main collection part for the blood. Blood bag label contains all the details from the donor including the blood group. Sampling bag allows pre-donation blood to be diverted and sampled. Clamps pinches the tubings to create a seal on the blood bag. The tubings allow the blood to flow between the veins and the blood bag. Needles and shield are used for the puncture to draw or deliver the blood, and the shield protects the needle and avoid injuries during the storage as well as transport. This is how a blood bag looks like. So there is a needle, a casing or the shield of the needle, various tubings and the actual blood bag. If you want to store whole blood, we will be using a blood bag called a single blood bag. If you want to separate packed cells and plasma, we use two separate blood bags connected with a tubing. This is called as the double blood bag. If you want to separate packed cells, plasma and platelets, we take 3 is called as the triple blood bag. At the same time, if you want to separate the packed cells, plasma, platelets and cryoprecipitate, we'll be using a quadruple blood bag. So this is how double, triple, single and quadruple blood bag looks like. So while donating the blood, we give the whole blood. But in case of transfusion, the whole blood or the components may be transfused. So apart from whole blood, you can transfuse the cryoprecipitate, packed RBC, plasma as well as platelets. So while you give the blood in a blood bag, won't the blood clot? There are chances, so which we will be preventing by adding something called as an anticoagulant. So what is the most common anticoagulant used in blood bags? It's called as CPDA or citrate phosphate dextrose adenine. Citrate acts as an anticoagulant by chelating calcium, and dextrose is needed for the metabolism of stored red blood cells. Phosphate lower acidity and have a higher concentration of 2,3-DPG and red cell phosphate. Adenine improves the viability of the red cells. Now, how many days does the whole blood or the packed cells can be stored? It can be stored up to 35 days under 2 to 6 degrees Celsius if it is collected in the CPDA bags. There is something called as SAGM also. SAGM is saline adenine glucose mannitol. It's an additive which is added to the blood bag, which will increase the shelf life of the RBC from 35 to 42 days. Then what is the capacity of a blood bag? It can be available in 350 to 450 ml capacities, and it can be stored either in a standing upright position in the basket or as a lying flat on the shelf. If you have any kind of traumas or surgery, we generally prefer whole blood transfusion. At the same time, if the person only need RBC, we take the blood cells and plasma separated by a process called as centrifugation and remove the WBC, which is called as leukocyte reduced RBC. Generally, this is done in case when a person is having anemia or surgery. Anemia can result from cancer treatments like radiotherapy and chemotherapy, blood cancer like leukemia, tumor production in the body which will inhibit the production of RBC, or because of low iron or vitamin B12. This can lead to low RBC as well as low hemoglobin level, which will lead for the patient to have an RBC transplant. Surgery can also result in RBC transfusion. Platelet transfusion is when you have whole blood and you collect the platelets and transfuse them to a person who is in dire need of platelets. So this one bag of platelet is called as a unit or pack of platelets. So this one pack of platelet can be collected from a random donor. This is called as the random donor platelets, where you take platelets from different people which have matching platelets, take around 6 to 10 units and combine them together in a single bag. Apart from that, if you want all the platelets to come from a single donor, you can take a single donor platelet by a process called as platelet pheresis or apheresis, in which the blood will be drawn, will be separated into its component by the process of centrifugation, and the needed component, that is the platelets, will be stored, and whatever is remaining will be given back to the donor. This is done with the help of a machine which helps in doing platelet pheresis or apheresis. Then who will need a platelet transfusion? It will be needed by a patient who is going through cancer, or by a person whose platelet count is low and is going to have a surgery. So due to chemotherapy or other cancer treatments, their bone marrow will not be working properly to produce platelets. Due to leukemia, in bone marrow, blood cells are crowded over by cancer cells. Low platelet count during surgery can cause high risk of bleeding, so such people will also be getting platelet transfusion. Plasma transfusion is you take the whole blood, divide them into blood cells and plasma, and the plasma is taken and is kept in a frozen condition. It is needed by patients who are bleeding as their blood is not able to clot, and cancer patients with disseminated intravenous coagulation or DIC. The last one is cryptoprecipitate transfusion, where you take the plasma, you keep it in the frozen condition, you thaw it afterwards. You see that a part of the plasma separates out, and it contains some of the clotting factors which is responsible for blood coagulation. You take this out, and this is called as the cryoprecipitate. 8 to 10 units of cryoprecipitate is needed for one transfusion of the cryoprecipitate. So far we have discussed about the structure of RH antigen, the genetics of RH antigen, blood transfusion, the different type of blood packs that we will be using, and the different types of transfusion like whole blood, RBC transfusion, plasma transfusion, platelet transfusion, as well as cryoprecipitate transfusion. Hope you understood the concept. Thank you.